Good afternoon, everyone. We are basically going to do the entire lab together right now. So it's great. I am your lab partner for today, and I'm going to guide you through how to do this lab because normally I would be actually doing this on the whiteboard, and since we can't do that, we'll try and see how this works. Okay, so I first of all want you all to turn in your lab to page 84. So we're going to work on pages 84 to 90 together. All right, so the first thing to do is to go ahead and maybe pause the video and carefully read the scenario that's listed on page 84. All right, so I'm going to go ahead and highlight what I think is incredibly important for us to understand what's going on. So the first thing to know is that we have a tetracycline resistant strain, and the bacteria is called Citrobacter frundi. The other really important information is that we have some E. coli, and it is resistant to sodium azide. So that means it's going to be on its chromosome. So what we want to do is allow these two bacteria to interact and see if conjugation can take place. So how are we going to actually do that? So for me, I like to actually draw out the full process, which you're actually supposed to do anyway. It says sketch out the process. So let's go ahead and do that. We've got two bacteria. We've got some Citrobacter frundi. And then we've got a strain of E. coli. Now, when I draw this out, I always like to draw out that the fact that they have a chromosome, so this is their chromosomal DNA. They both have chromosomal DNA. And then what's the other important information is that the Citrobacter frundi has a tetracycline resistance gene that may or may not be on a plasmid. So really what we have going on here is we've got a plasmid, potentially, in Citrobacter frundi that we think may have an antibiotic resistance gene. And we think this is TET resistant. Yeah. Go ahead and write that in here. All right. And so what do we know about E. coli? Sorry, that's my phone ringing. What do we know about E. coli? We know that it has resistance to sodium azide. So we're going to draw that in as well. We have the antibiotic resistance gene that would be potentially here. The other strain has sodium azide resistance. All right. So the question we're really asking is it possible for the Citrobacter frundi to extend out that sex pilus and transfer the tetracycline resistance gene? So if that happens, again, this is going to get replicated, and then a copy of that will be transmitted to the E. coli. So what we would end up with is basically E. coli. So what we would end up with is basically E. coli that was resistant to sodium azide and the tetracycline resistance gene. So we'll just call this E. coli prime since this is our trans conjugate. So now the second question on the lab handout says, how would you select for trans conjugates? So one way to do that is if we can grow these bacteria in the presence of both antibiotics, both sodium azide and tetracycline, then we'll kill off the parental strains. So the addition of sodium azide is going to kill off the Citrobacter frundi, and the addition of tetracycline will kill off the initial E. coli parental strain, such that only the E. coli that underwent conjugation is going to survive in the presence of both sodium azide and tetracycline. So that's how we're going to select for the trans conjugates. All right, let's go ahead and turn to page 85. Okay, page 85 talks about why we have all the different controls that we do. And so I'm actually not going to answer these questions for you. You should take your time and answer this on your own. It's incredibly important, but I want you to rationalize your way through, and I think you've got enough information on there that you can go ahead and do that alone. In order to answer the questions on page 86, you should compare page 86 to page 83. And in the previous video, I thoroughly explained how to answer those questions on page 83. So please go back and look at that. Based upon your comparisons, you should be able to easily identify the 3 control groups that are missing. All right, so now let's move on to page 87 together. All right, so when you are looking at these experiments, and you should be able to systematically do this, you can anticipate some questions like this on the lab practical. So what we've got here is a proposed experiment, and what we want you to do is to look at the results and see what is of the controls, what didn't quite work correctly, such that we aren't fully able to determine whether or not conjugation took place. So go ahead, pause the video, take a minute to identify where you think this experiment went wrong. Based on the production of colonies and where you see lawns and no colonies or countable colonies, if you look at all the controls, they actually seem to work in this experiment. The only thing that didn't work was actually the JM109 with HY842 with both antibiotics. We see no colonies. If it worked, we should have seen countable colonies, but we did not. So you actually could conclude here that no conjugation took place. Based on the conditions provided, the conjugation did not happen, and but we can conclude that because all of our controls actually worked. All right, so let's do the same thing with scenario number 4. I'm not sure what it says, scenario 3, but go ahead, pause the video, take a minute, analyze it, see what controls maybe would cause you to think that something didn't work, and then make your conclusions. Did conjugation take place or not? Hopefully, upon your analysis, you recognize that here, the JM109 appears to not be resistant to the ampicillin. So anywhere where we've got JM109 and normally it should have been resistant to ampicillin, we really can't conclude what's going on. So here we can't confirm whether or not conjugation took place because our super important controls would indicate that the bacteria was indeed not resistant to ampicillin. Here, hopefully, once again, you notice that we had some issues with that JM109 strain. Normally, with zeaxanthin, we should see no colonies. However, we saw a lawn. So that would indicate that the JM109 either is resistant to zeaxanthin, if the zeaxanthin is working, or perhaps our zeaxanthin has degraded and that antibiotic is no longer effective. So really we can't make any conclusions about whether or not conjugation took place because the JM109 strain is showing resistance to the zeaxanthin. The final page in your lab manual is page 89, and we've already discussed what the results should have been for this experiment. I will just quickly remind you here. So if we are looking at the JM109, again, this should have had the antibiotic resistance gene on a plasmid, resistance to ampicillin. So again, anytime ampicillin is present, it should have been fine. You should have seen a lawn. If there's no antibiotics, we should have seen a lawn. However, in the presence of zeaxanthin, we should have seen that antibiotic was incredibly effective and it would have killed off all the bacteria, so you should see no growth. And the same is true here because zeaxanthin is on the plate. All right, for the HY842, it's not resistant to ampicillin, however, it is resistant to zeaxanthin. So anytime zeaxanthin is present, we should have seen growth. You'll see a lawn. Again, no antibiotics would have a LON. There's no selection against that bacteria. However, adding the antibiotic ampicillin should have killed off the HY842, and the same is true here when you're using the combination of antibiotics. All right, so in the final scenario, what we have again on the plate is both JM109 and HY842 as well as the trans conjugates. So all I've drawn here are the trans conjugates, which happens very rarely. So when we have no antibiotics present, nothing is being selected against, we should definitely see a LON. When we add a combination of the two bacteria along with the trans conjugates, we're going to see a LON in the presence of zeaxanthin. Again, that is because we are killing off all the JM109. However, the HY842 parental strain is still in there, so we're going to see a LON. When we add the ampicillin, we're going to be able to kill off the HY842. However, the parental JM109s will survive, so we're going to see that. Only when you add the combination do you kill off both parental strains and only select for the trans conjugate. So that's going to be the only scenario under which we find countable colonies. All right, so that is the end of your lab. You do not need to do the one-page analysis of your results. Fantastic job. It was a pleasure doing lab with you today, and you will find further instructions posted for you on Canvas. All right, take care.